There we go. And I'll really focus on this idea of the tools that we've built in the OME project, really trying to find ways to make data so-called fair. Some of you will recognize that acronym. I'll tell you more about that in a second and define what that means. You can think of it as just making data truly accessible. So it's winter here in Dundee, and what you can't see is there's about 1.5 feet of snow on the ground, and we're all stuck in our houses because we can't get out. So it's not only COVID that's locking us down, but actually the snow. This is a picture of Dundee at midnight in June. And so there's a lot of light. We're fairly far north, and sort of the bottom left-hand corner here is the research center at the School of Life Sciences at the University of Dundee, and that's the home of OME. So without any other ado, I do have the chat window open, and so if you have any questions, you can type in there. Happy to answer questions during the talk. So first of all, I know it's a very international audience, so a bit of a disclosure. As I mentioned, there's a commercial arm to OME called Glencoe Software. We founded in Glencoe in about 2005. It's been running ever since. And basically, the idea was to provide a commercial access for commercial organizations, for example, biotech and pharma, to the open source tools that comes out of OME. So this is really just another way to get our technology out into the community. So just by way of disclosure, if anybody has any questions about running an academic and a commercial project, I'd be very happy to answer that. So this slide sort of summarizes the problem that we're working on, and my guess is that many of you are working in some of these imaging modalities, maybe several of them. You know that imaging is everywhere in the life and biomedical sciences. That's not just because we get to make pretty pictures, although that it is nice. I get to show you, if you're an imaging person, you get to show nice images in your PowerPoint presentations. But really, it's because we have a spatially and temporally resolved measurement of the structure and the composition and the dynamics of biological systems across many different scales. So you will know that in electron microscopy, we're really looking at the submicron scale. We have the facility to label biological model systems and organisms in many different ways. So we can probe the dynamics and the behavior of molecules in individual cells. In whole animals, we can use those tools for drug screening. Certainly many of you will have seen medical imaging, MR, etc. And most recently, the pathologists have really gotten into the act, and pathology is undergoing a revolution, changing into a digital imaging science as well. So yeah, imaging is everywhere. And what's important to remember is that these images, as I said, pretty pictures, but are fundamentally measurements. So the pixel values will be somehow faithful representations of the spatial relationships, but also, for example, especially in fluorescence imaging, the relative concentrations of different molecules. Obviously, examples of dynamics. And so these measurements are quite accurate. Some of you will, I'm sure in the course, you'll talk about how the microscope is a linear operator. So it's faithfully transmitting these relative concentrations and the relative positions in a linear mapping. So we can actually depend on those measurements and do subsequent calculations on them. And obviously, we don't take a single image of a group of cells or a single time-lapse sequence, but we now build collections of these data sets to get the statistical sampling that we need. And so it's pretty common now for every paper that uses imaging to have a whole collection, many data sets or many collections of data sets, that look at the biological or the biomedical system under question. And so that starts to raise a third type of use of this data, We talked about pretty pictures, we talked about measurements, and that's really this idea of a resource. So fundamentally, perhaps in my lab, I'm collecting images of time-lapse images of cells going through the cell cycle, and maybe you have, a really, important new tool that you've developed for analyzing those data. In this internet powered world, it should be possible for us to exchange, for example, data and algorithms, and for us to be able to share data and share algorithms. And so the idea of a data set being a resource, either in that kind of form of a collaboration, or for example, like many other fields do, when you publish a paper, the data set's being published alongside the paper, really is starting to come to the fore. And so it's really this idea of doing measurements at scale and building data resources with these imaging data across all of these different modalities and all these different scales that's really been driving us forward. And that's really what I'm going to talk about today. Okay. So this project I'm telling you about, OME, is an open source project. Everything we do is publicly available. In fact, we don't do any private support of institutions. All of our support is online and public. And what the project aims to do is to build the software technologies that really make it possible to bind all these different functions together that you see on the periphery of the slide. So, for example, many of you in this course, you will see many examples of this. Lots of new imaging modalities and huge array of different types of image acquisition systems, lots of different analytic tools. So what OME is trying to do, and our definition of success is when it's possible to take a pre-existing analytic tool that perhaps was built against data from one imaging modality, and apply that tool to a much broader range of imaging acquisition systems or domain types than the tool was first built for. Put that another way, a new imaging modality comes online, and it can immediately be accessed by the very broad range of tools that exist, of analysis and processing tools that exist out there in the community. So this is really all about this word that you'll often hear, so-called interoperability, this data exchange or exchanging information across all of these different functions. Some people would call this the central broker. You could call it the glue, whichever word is most useful to you. But what we're trying to do is build the software tools to make this routinely possible, and also to be able to work at scale, and particularly at the scale of the type of data sets we're now collecting. So that's really the goal of the project. As you'll see, as I kind of go through the slides, we've been broadening out and doing a lot more in the way of analytics, high-end processing, and some visualization. I'll show you examples of that as we move forward. Okay. The URL for the project is there, openmicroscopy.org, and you're welcome to use or abuse in any way you wish the technology that we release. So let's talk a little bit about that technology. I won't tell you too much about this. Again, it's all available, it's all open. Happy to get into much more technical details as we go on. But I'll give you kind of a brief overview of the things we do and how we use them. So these, as I said, this is an open source software project. It comes out of an academic lab, completely grant funded, and you can think of these as the products or the pieces of software or data specifications that we build and release to the community. And so there's several names here, and I'm not going to tell you about all of them today. I don't have time, but I'll focus primarily on bioformats, OMERO, and IDR. Now, bioformats is a file format conversion library. It's a plugin library that reads lots of different data in lots of different formats, and this is very important in real time. As the data is being accessed, converts that data into a common model so that lots of software, open source software, commercial software, and others can use it. So why do we do this? Well, many of you will know that your imaging systems, especially those from commercial imaging vendors, write data in proprietary file formats. They write the binary data and critically the metadata into a proprietary file format. And so what Bioformats is doing, what we do in the Bioformats project is reverse engineer those file formats and build readers, open source readers for each of those formats. We embed them in the Bioformats library. And so what Bioformats does is recognize which of the many different formats your data is in, and then in real time converts it into a model that this other software can understand. This is really a community project, so Bioformats, the success of Bioformats comes from contributions from the community. So we hold about, on the order of about 600,000 files that have been donated to us by the community. And we've used those files as test cases, and in fact we use them as a testing system. So every day Bioformats is tested against this library of file formats several times. And so one thing that a lot of people don't realize is the Bioformats development project is actually a high performance computing project, because we have to do this, effectively do all of this testing several times a day. Now Bioformats is a real success story for us. It's used everywhere. I'm pretty sure most people who use Bioformats don't even know they're using it. It's embedded in Fiji. It's distributed with lots of different software. Several commercial vendors include it in their own software. And so it's been a great success for us, a great opportunity to interact with a wide range of community. We can we believe there's something like 40,000 to 50,000 IP addresses worldwide that are using Bioformats. If you think about how firewalls are constructed, to us, your institution is a single IP address. So we just see a single IP address using Bioformats. So we think there's hundreds of thousands of users that started over 100,000 times a day, every day. So that gives you some sense of the success of this library. Now, the second tool I'm going to tell you about is OMERO. OMERO is a full-blown data management system. This is all published, by the way. So if you want to find more about the technical details, do please look at the paper or look at our website. But basically what OMERO is doing is taking many different types of data, for example, imaging data, metadata around ownership or maybe analytic results, search indices, etc., and using technology that's now actually pretty standardized in the world. There's a middleware application we call OMERO Server that's connecting all of those different data types and amalgamating them in some way, connecting through a secure internet connection out to a client application. Now, you probably could realize that in science we can't define what technology you would use to access your data. So these clients we have to be able to support clients in many different environments. So several programming languages, for example, C, C++, Python, scripting languages like MATLAB, Java, a web framework, etc. And so without going in the details, we have the technology to be able to support all of those simultaneously. Now, I want to emphasize that what you're seeing here is open source software. We built this with the intention of building a tool that laboratories and institutions could use to handle their imaging data. We didn't intend initially for this project to be about storing and serving other people's data. We have started doing that, and I'll tell you about that in a second. But most of the OMERO usage is around institutions worldwide that are downloading the open source software and implementing it in their own institution to manage their own data. I should emphasize again, what you're seeing here is a server-client environment. The server-client environments are what we're all using. We're using it right now. Zoom is a server-client environment. There's a server handling all of the video compression and everything and feeding out to your client application. Server-client environments are how you read your email. It's how you read the newspaper. That's what WhatsApp is, and so on. So in some sense, all we're trying to do in the OMERO project is take this technology idea that is so well embedded in everything we do and just apply it to everything we do in bioimaging. I think the last thing to say is that I mentioned many different clients up here. So we have a lot of examples. So there's this connection from the server out to the clients. It's called the API, or the Application Programming Interface. This is open. And so the tools we build all work, but lots of people build their own tools. And so you can do that, for example, using these different programming languages. So there's a lot of flexibility built into the system here. So with bioformats in OMERO, here's how we end up structuring, how we handle data with these tools. So if you think about a standard imaging facility producing, usually tens to hundreds of gigs, sometimes terabytes of data, many different modalities. Most institutions will have that writing onto some sort of large data store or maybe multiple data stores. So bioformats, remember, is providing that real-time translation of all of the different file formats that are coming out of that facility into a common model. So you can think of bioformats as an interface, sort of a window into all of those, or a single access method or a single access point into all of those different data types. And then we take OMERO and the OMERO server, and we put it on top of that. And this is sort of another interface or another API that provides you access to all of this different data you see on the left-hand side of the slide. So for example, that access might be in a web browser, sitting on your computer. I used to say in your lab, your laptop in your lab, but now it's your computer at home. Alternatively, the same interfaces can be used by large analytic and processing tools, for example, cores and clusters for handling data at scale. So the next couple of screenshots you're going to see are actually from a web client on my computer, and you're going to have to just imagine that what's sitting underneath that is effectively this whole system, including OMERO and bioformats, but obviously the data stores as well. So here's a screenshot of what we call OMERO Web. It's a web client for accessing this data. Sort of has this left-hand panel, middle, right-hand panel arrangement. The left-hand panel has file organization. The middle is thumbnails and images, and the right has metadata. You see some analytic metadata coming out of, as it turns out, Cell Profiler in this case. So that's OMERO looking, in this case, at high-content screening data. But because we have bioformats, exactly the same system can look at, for example, whole slide imaging data. This is from the TCGA data set from the NIH in the United States. And so these are very large images. You can see, I hope you can see on the slide, the pixel sizes here, roughly 100,000 by 40,000. So we can handle that kind of data. But note that the metadata here is very different. And so OMERO has ways of adapting to the different types of metadata that are handled in different experiments. And this is what you're seeing here is the data that comes out of the TCGA data set. These are the analytics that they have produced. And a final example, again, this file organization thumbnails, these are obviously human MR data. And some of you will recognize the metadata here as DICOM tags. So again, this adaptable metadata system can handle many different types of metadata. Okay, so I took out a lot of the slides around analysis. I think you're probably seeing that elsewhere, but because we have this API, this open API, OMERO can be, as I mentioned, can be addressed by many different analytic tools, segmentation tools, some open source tools, Cell Profiler, Elastic, Fiji, all of those names. Many of the deep learning tools that you've heard about, TensorFlow, and so on. So again, the idea of this common interface can be accessed by many different things. But for the rest of the talk, what I want to get to is really this idea of sharing data and doing so at scale. So the reason that we can do this is OMERO has a database inside it, and databases do lots of things, but one of the things that databases do is control access. They decide if you can access the data that you want to access and if you can access anybody else's data. Now, a simple example of that is your bank account, or maybe when we used to travel, maybe your online, your airline reservation system. But each of those have a database underneath them. And so when you log in, you see the information related to you, but you don't see, for example, your neighbors, you don't see mine. And OMERO is using exactly the same concepts. But we can adjust these depending on what you want to do. So for example, we have this idea of groups, and groups can have many different status. So you and I can be in a private group, so I can see and annotate and write my data, but you can't see mine, vice versa. Not very collaborative. Some people like, sorry, a very huge chunk of ice just fell off of my house. So I apologize for the background noise. That was very scary, actually. Um, so, this idea of we can use these idea of groups to moderate how different members of the group can work with the data. So we can have a read-only group. So I could read, I can see your data, you can see mine, but I can't do any annotations or analysis, and I certainly can't change the data or write the data, and vice versa. You can't do that to my data. Or maybe you and I can analyze each other's data, or maybe we have full permissions and we can all do anything we want with each other's data. Similarly, and this is where the data sharing gets a little bit more interesting, you can set a group status so that, for example, data is public. Data is available as a URL. And you have those same permission concepts. Data can be read-only. That's usually what you want to do in the case of a public resource, or read and analyze, or even read and write if you so choose to do that. So what do you do with that security model? So here's a paper that we published. I'm not telling you anything about the science in that paper, but this is a paper we published a couple of years ago. And if you scroll down, if you go to this DOI and scroll down to the data availability section, you'll actually, and click on we have a DOI that's in there. So a DOI is a digital object identifier. It resolves to our public OMERO instance here at Dundee. And this is what you would see. You would see the original data associated with the paper in those figures. And you can see they're organized according to different figures. We have a figure making system application called OMERO Figure. If you want to find out more information about this, there's lots of online videos. Just Google omero.figure. And so you can take the data in OMERO, build a figure, do all the annotations, etc. And we have all of the figures published as well. So that's one example of publishing data. So that's our lab publishing data. But what about thinking about institutions? And so here what I'm going to do is take you on a quick tour of sort of making data available at the institutional level. So this is out in Portland at Oregon Health Sciences University. This is Joe Gray's group. They have several projects where they're doing large-scale profiling of human tumors. This is a case where they're looking at responses to to drugs in the context of various extracellular matrices. There's a large-scale screening experiment, and all the data is handled in OMERO and published with OMERO. Back here in the UK, this is at EBI. This is the EMPIAR Electron Microscopy Archive. Now note that you're going to see this looks very different than anything I've shown you before. Obviously, there's images inside. So OMERO server is sitting underneath because of that client-server architecture. Very, very different looking clients can access the same system, same technology. This is at Harvard Med School. This is Peter Sorga's group. So Peter's group has really been developing and pushing this technology called cyclic immunofluorescence. So this is profiling tissue sections in this case. So this would be a thin section, probably 10 or 20 microns. But now not doing an H&E stain, just to show you, for example, proteins and nuclei, but in fact this has about 40 different markers. So this is immunofluorescence, taking a large number of different markers on it. The yellow signal is cytokeratin, and so this is from a breast cancer patient. And so Peter, with his colleagues at Harvard Medical School, have been developing technologies to effectively use these tools as companion diagnostics. But again, sitting underneath this is this data management system, OMERO, making this data available to pathologists all around the med school. This is, again, in the US. This is at Vanderbilt, publishing images associated with human pancreas data. So these tissues are very precious. And so making the data and the staining and the results all public. This is in Japan. This is Shuichi Onami's group at RIKEN are building a data repository for the Japanese community based on OMERO. So those are all OMERO sitting underneath, serving imaging data and metadata to all of these different applications. Another example back here in Dundee, so this is pretty much vanilla OMERO with a few tweaks, and we're actually using this for a online training for anatomy and histopathology. And so several hundred students every year use OMERO for accessing these tissue samples. And it turns out this is a lot easier than having to have slides for all of the students. So this gets to this idea of making data fair. And so I want to define fair. Some of you will have heard of this, some of you might not have, but so-called findable, accessible, interoperable, and reusable. And so the idea, and this idea comes from a paper published by Wilkinson and colleagues a few years ago, is that these data sets that are coming out of our scientific experiments really must be available for the community. They have to be found. You got to be able to find them. So remember that DOI I showed you. Once you find them, they have to be accessible in some way, that there's an open API that connects them, that makes the data available. That there's an interoperability idea that you can connect many different ways of connect many different applications and exchange metadata that can inform different functions. And they also have to be truly reusable, so properly licensed, etc. So that's really what's inside each of those examples, is us providing the foundation to implement this concept. Okay. So in the final part of the talk, I want to take that idea of sharing data and then push it a little bit further. So I showed you several institutional resources that are using OMERO for publishing their data. And that, so we've been working on that for several years, and that really raised the idea that we would it be possible to think about making truly a centralized public resource for bioimaging data associated with publications. And so many of you will know of resources at NCBI in the United States, or at RIKEN in Japan, or at EMBL, at the European Bioinformatics Institute, which are online resources, for example, genomics data, and certainly structural data, the PDB, and so on. And so, those are extremely well established and now references for the community. And the question was, could we think about doing something similar for bioimaging data? And I have to say, we worked on this problem for several years, and there were a lot of statements like, well, the data is too big, it'll never work, and things like that. I can tell you we've solved all of those problems. And we're now really, with this project that I'm going to tell you about, really the question is how we do the annotations and how we do the curation, how we generate the most value. And so that's what I'm going to tell you about in the next few slides. So this project is called the Image Data Resource, or what I'll call it the IDR. And this is sort of a quick tour of the IDR. And so the idea behind the IDR was that we would take many different data sets from many different domains. We would integrate them inside an OMERO system, capture as much of the metadata as we could, the annotations, analytic results, etc. Make all those publicly available in a web-based interface, so people anywhere in the world could access them. And so we would have search implemented, etc. But critically, and this is going to become more and more important as I go through these slides, we would allow data download because some people want to download the data. But if you think about downloading terabytes of data, that probably is not something you're going to routinely do. So we have to actually start thinking of smarter ways to provide access to the scale of the data sets we're talking about here. So either we bolt on an analytic framework, a cloud-based analytic framework next to this resource in some way, or we provide a download capability or an access capability that doesn't force you to download 5 terabytes of data when you want to see these data sets. So that's what I'm going to tell you about now. So we've done all of this, and this is now up and running. It's been running since about mid-2016, and the URL is there. I should say this is running at the European Bioinformatics Institute, or EBI, in Kingston. We're very, very grateful for them providing what they call their embassy resource. It's a cloud-based resource that we've been able to use. And we simply, as I'll tell you in the next few slides, we simply could not have done this without that collaboration. We're very grateful for that. So these data are a little out of date because there was another data release this morning. But this gives you an idea of the scale that we're operating on. The raw data size now is over 200 terabytes of data. There's approaching 30 million image files. What we call five-dimensional images, so space, time, and channel, so all of the structures that we work with assume that a single image has 5 dimensions, so 3 spatial dimensions, time, and frequency or channel. So over 10 million of such images. If you look at 2D planes, that's I think now over 75 million 2D planes. Large number of different organisms, about 83, I think up to 85,000 genes now, large number of antibodies, and actually a series of chemical compounds. So lots of imaging data and then lots of metadata is probably the way to think about this. IDR is heavily used. We get about 20 to 30 million hits a year, about 50,000 unique IP addresses worldwide, which seems to somehow map to a number that's around the number of research institutions in the world. So that's great. More importantly, what is IDR doing? So we are publishing the data sets associated with peer-reviewed papers. So papers are coming out in the journals that are listed there. And then alongside, there's a data set that is published that is available. And so, for example, you see an example data availability section here from a paper. There's various data sets associated with this paper. The imaging data have been submitted to the imaging data resource. There's a URL, and then there's a identifier that gives us that provides a way to access that data. So I would emphasize, you think of the number of data sets that we've published in, since around mid-2016. So coming up to our 4th birthday, we've only published about 90 studies. And you might think, well, 90 is a lot, but if you think about all of the papers that have been published since that time, it's clearly a much greater number. So why aren't we published? Why isn't this number larger? And that really gets to this idea of so-called reference images. So in IDR, we're focusing on many different imaging domains, all the ones I mentioned before. But what we're looking for are data sets where we have a lot of annotation of, for example, genes, chemical compounds, biological information, etc. So these comprehensive studies, because that means we can capture a lot of metadata that defines the different treatments and provides us a way to connect the data, to connect these data sets across different domains. Where possible, we want to get phenotypic information, any ground truth information, any analytics, etc. And so these are very, very rich data sets. And so so-called reference images. So these are a small number of data sets which are going to meet this category, meet this kind of criteria. And so there's a whole process by which we evaluate submissions and select those which we think we could get this very rich metadata to put them into the system. Now that does raise the question of what about everything else? As I've said, as I've indicated, reference images by our criteria are a small minority of the total data sets that are being published. And so for that, we've been working with our colleagues at EBI, and EBI is building what they call the Bioimage Archive. And you can think of the archive as the very large resource or repository for all of the other data. And the ideas here, all the other bioimaging data that is not reference metadata, I should say. And so the ideas here were published a couple of years ago. This paper is open access, and you should be able to see it. You can look at that. So at least in principle, what we foresee is that as we're building this, what we call this bioimage ecosystem, that most of the papers that use imaging would be publishing their data in the archive. And to make that publication routine and quite rapid, there's probably a minimal metadata specification that the archive would use. And EBI has led the way in defining that specification. That will hopefully be published quite soon. There's a paper in revision. Um, for those studies which are reference data, then then there's a specification that's a little bit more that will be a little bit broader, so-called reference metadata specification that would come into, for example, the IDR. And so what we've started doing now, so all of this is not just a PowerPoint slide. This all exists and is running. So I'm telling you mostly about the IDR, but the archive is also running. And so these pathways do exist now. And so if you have data associated with a publication, as you're entering the publication process, and ideally, during the review stage, starting the submission process, contacting either the archive or the IDR is definitely a great idea, so that the process of submitting the data, metadata can happen. So it's not just us, it's not just these two projects that are doing this, is that we actually see this as a much larger effort. And so with our colleagues, Shuichi Onami at Riken, Shuichi is taking these ideas and really building on them for Japanese national researchers, and he's built what he calls the System Science Biological Database Repository. So that is an archive for Japanese researchers doing bioimaging, as well as an added value resource where we're doing a lot of this additional metadata curation and annotation. Okay. So it's actually a huge effort, and we're starting to see other national efforts coming online. And so I think, over the next 5 years, you're going to see on both sides of this diagram, you're going to see more repositories appearing. That raises a lot of questions, and I'm happy to take that into the discussion. I hope you'll understand I don't have all the answers yet about how all this will work, but what's really gratifying is, so it's now 2021. In 2015, when we started all the work, none of this existed. So in 5 years, we've been able to build all of this, and so progress is moving quite rapidly. So let me tell you a little bit about what's in the IDR. Lots of different imaging modalities, and so these are just a few of the examples from many different papers. You'll recognize super resolution, histopathology, phenotypic screens. This last one is so-called geographic screen. This is the Tara Ocean study. So this was published a couple of years ago, and this was a sampling of the world's oceans for plankton. So to put it simply, put a bucket in the ocean in different locations all over the world, do a full metagenomic analysis to identify those species at a molecular level, but also stain and image all of the species that are discovered. And so we have this record of incredible number of species in the world's oceans in the IDR. Other examples, this is osmotic response. This is a single cell profiling experiment. So this is RNA fish, and so for every image, we have obviously the images. We actually have all of the ROIs that the authors used to analyze these data. I took the slide out, I apologize. That is an example of data that's going both in the IDR and the image archive, so the same data set in both places. This is an example of from Geneva Farm. This is from Philip Keller's lab. This is the definitive, this is light sheet microscopy, and this is the definitive fate map of the mouse embryo going through several hours of development. This is from Matabushi's group in the United States. This is a deep learning study where they are using their AI technology to discern between biopsies, normal and damaged heart tissue, biopsies of cardiac biopsies of normal and damaged tissue due to ischemia. And I apologize, it's probably a little bit small, but on the right-hand side here, you may be able to see the metadata around the case. Obviously, all this data is anonymized, but there's data around the case and the progression of the disease. Many of you will have heard about the Human Protein Atlas, an amazing project out of the SciLife Lab in Sweden. So they've developed a library of antibodies recognizing the most of human antigens, and then stained cells and tissues with those antibodies. We have all of the tissue. We are getting all of the tissue data, I should say. We currently, I think, we've published 9,000 of the antibodies, and so that's several million images where we have the original image that's available and the staining and critically all the annotations around the case as well. Oops. Um, it's a tricky time for all of us. It's the SARS-CoV-2 pandemic. Hans Glaver's group has had a very nice paper in May in Science showing the infection of human intestinal organoids with the virus. We have the these are very large EM maps, hundreds of thousands of pixels on a side. All of that is viewable in IDR. We are cross-referencing. This is an example of cross-referencing the data. So our IDR metadata cross-references the EMPIAR archive, and EMPIAR is the EM cellular imaging archive run from EBI. They are cross-referencing us, and so that the raw data, the files are available in EMPIAR, and they can cross-reference to us. So we provide visualization of these large maps. They're providing very nice download specifications. And so, I showed you lots of examples of imaging data, and so really, what's the point of doing all this? Why do we have all of these different data sets? And so what I'm going to show you are some examples of using the metadata in various ways, hopefully to provide a useful insight into what the data is telling us. So this is some image in the IDR. In this case, it's an siRNA knockdown of a specific gene. The authors submitted the imaging data, a lot of other metadata around the siRNAs that were used, obviously the cell line here, but also the analytics and effectively their interpretation of the analytics. So what they saw in this case was an elongated cell. We can actually, we can use these terms, so you will know that genes have a defined namespace, and so we can use that. But there are now several ontologies that capture, for example, phenotypic information, and so we can look for and identify terms that in this case, the Campo ontology, which capture that concept of a cell elongation. And so that gives us the chance of linking elongation into a gene name. We can build search tools on all of this, and so you could search IDR for genes or phenotypes. Those phenotypes can be text or they can be IDs. All of that is there. We can then integrate, for example, we can ask IDR, okay, tell me what studies you have for around those that elongated phenotype. This shows 3 of the studies that would come out if you did that query. This is all published, so I won't spend a lot of time into it. Looking across those studies, we can build gene networks across the different genes that are associated with those phenotypes. And it turns out we were able to hypothesize that there are novel systems controlling cell shape in, for example, POMBI, but also human cells. Again, I'd refer you to the paper. And the point, I guess that's, I showed you one story about phenotype. What I want to emphasize is that we're annotating with a large number of different ontologies, capturing many different concepts, some around, for example, the organism, the imaging modality. As I mentioned, phenotype, that's the cellular microscopy phenotype ontology or Campo, clinical status, and what I'm going to focus on the next couple slides is chemical compounds, which have turned out to be quite interesting for us. So we have these data sets coming in, and then the process of working with the authors to identify which ontologies we need to use, what annotations we need to make, is really what that added value resource that the IDR can provide. It's work, definitely is work, but hopefully it generates some value. So here's an example. So there's an image in one of the studies. This is using a compound called olaparib. Some of you will know that compound. And what you're seeing here are the various IDs that are in the community for identifying that compound. So some of you will know that public resource called PubChem has an identifier. Kemble has an identifier. There are SMILES codes, which is basically a way of building up the structure, INCHI and INCHI-key. And so we've been able to capture all of these. This, again, is a lot of work, but we can do this, and we can do it at scale for thousands of, for the many thousands of images that are coming in. And so what that allows us to do is to build search facilities based on, for example, these compounds. So if you go to IDR and you search by compound, you can search for example, for remdesivir. And what that will do is take you to a study that was submitted by Ellinger and colleagues. The paper is still in press, but they've asked for the data to be published. And this is a dose response curve of remdesivir in human cells. So their paper is presenting one of the first studies of SARS-CoV-2 infection in human cells. And you can see that as they lower the dose, they're actually getting effectively loss of cells, a cell killing event here. Now, many of you know that remdesivir, there was a lot of excitement around remdesivir in the early days of the infection. And this kind of data was very useful. This shows that remdesivir is actually blocking infection. Probably clinically, this is less valuable because you have to get remdesivir into patients much earlier than patients are showing up in hospitals. I included this slide just kind of just as slightly entertainment. You might have asked, what about hydroxychloroquine? We do have that data in the IDR, but you will not be surprised to show to learn that that didn't actually have any effect. So in any case, we're building the IDR. We've had some success here. We started to see, and I've shown you examples of cell and tissue data, and so we've sort of divided IDR into two different domains, same data sets, but your search results are biased, whether you're coming into tissue or cells. We think that might help users. We've built a whole analytic environment next to this. We call this the virtual analysis environment, or VAE. The URL is at the bottom of the slide there, so have a look at that. Some of you will know this technology. This may be new to some of you. This is a Jupyter notebook, which we provide sample notebooks for in the virtual analysis environment for doing analyses. This is a query where we're querying IDR for gene names on the left-hand side here and asking for phenotypes. So we're asking IDR, what phenotypes do you have associated with each of these genes? I showed you an earlier example of one phenotype in one gene. This is, some of you will recognize this is the ARP23 complex. We're asking what phenotypes are associated in the IDR with perturbing the ARP23 complex. And so what's interesting about that is that if we ask IDR, okay, tell us the gene, if we ask IDR, tell us the phenotypes associated with genes, and then take those phenotypes and say, tell us the genes associated with those phenotypes, this is an example of what we get out of that. So these are gene names. This is, again, ARP23. And the horizontal axis here are the genes that IDR says have similar phenotypes as ARP23. We take those gene names and then we plot, and then we then query string. The string database is the definitive online resource for co-expression, gene co-expression, biochemical interaction, and other evidence of interaction between gene products. And what's interesting is that the significance of these interactions, so string already knew that several of these genes are already interacting. So we haven't discovered anything with IDR here, but this is an interesting validation experiment. Just by picking up gene names and phenotypic annotations, we can recapitulate a lot of other information that's known in the literature. Obviously, we're taking this forward. I should say that for those of you from NCBS, this was done by a colleague of yours who joined us in Dundee for several years. He's now moved on to AstraZeneca. So we think that these notebooks can be provide a really interesting way of analyzing this data. We're working with our colleagues at EBI to scale this up using a technology called Binder. This neat, here we're taking notebooks from the Allen Institute for Cell Science, from Susan Rafelsky's lab, integrating commands to integrate them with the IDR and publishing all of this. I'm happy to talk more about this. This gets very technical very, very quickly, but this is a really interesting way of sharing data, analytics, and making them available. Finally, I'll just say that for those of you who want to build an IDR, I've said several times, all of our code is open source, but building the technology stack, all of the software stack that runs all of this is actually quite a complex operation, but we've automated all that. So there's scripts, effectively these software scripts that build an IDR system for you. That's using a technology called Ansible. Again, some of you may not know what this is. It's okay. This is all open source, and so now anybody can build their own IDR. And our idea there is that there should be not just a single IDR, we're not the only ones, but hopefully multiple IDRs. And we really want to enable that capability. So I showed you our work on the cell IDR and a tissue IDR. There's now work starting up on IDR for the human cell atlas. There are digital pathology IDRs. There's a marine biology IDRs. And we know there's others under constructions. As I told you, in some way, all of this is going to have to integrate with archives, perhaps the bioimage archive, and all of that's still to be determined how that will all work, but that's the direction we're going. Okay. So just to summarize the section of the talk, I think we've shown that we can make these data publicly available, do all the curation, take data from the community, work with submitters to make all of that happen, make all of this scalable, and also enable reanalysis, and also show that other people can do this as well. And so that's really, we think that's a sort of a success story. We're very proud of that. Again, just to say, all of this is open source, and everything is on GitHub, and you should be able to find it. But if you need URLs, just let me know. Okay. Now, where is all this going? And so in the final section of the talk, I'll hopefully give you an idea of what we're focusing on now, and where we're taking this. And in fact, where we're focusing most of our efforts is all the way back in file format. So I showed you all of these applications, but in fact, running IDR and running at the scale that we're working on is really exposed us to the challenges of handling all these file formats, and it raises this question of what to do with this bioformats library. So here, I mentioned bioformats is a real-time converter, and so there's lots of different sources of different proprietary file formats, many different companies out there. And bioformats is now sitting in a wide variety of different software, serves as a plugin to pull that metadata out and present that to the application. And as I hope I've convinced you, that seems to be working very well, and many of you probably use this routinely. Some of you know it, maybe some of you don't. But the fact is that there's always more and more vendors, and there's more and more file formats. And this gets to be sort of a difficult task after some time. And so for a few years ago, we started looking at the number of truly new file formats we were seeing, and starting about 2014 to 2015, this started tailing off. And so we started publishing, so if you go to blog.openmicroscopy.org, there's the URL. We started publishing statements which were saying, this is just crazy. This doesn't make any sense. To put it very bluntly, getting hard-won grant funding to reverse engineer file formats is not exactly the easiest thing to do. Frankly, grant funding agencies much prefer to fund cool science than proprietary file format reverse engineering, okay, as you can imagine. Although I have to say we've been reasonably successful, there has to be a smarter way to work. But that argument is a little bit about the file formats, but is ultimately about the directions that the whole field is going. So many of you will have at least heard about, probably started to access light sheet microscopes, perhaps focused ion beam scanning electron microscopy, where the data sets are getting into the terabyte range. And we have these multi-dimensional images, which are actually quite unwieldy. And the fact is that the file formats that we have access to in sort of the routine bioimaging world are fundamentally plane-based or two-dimensional based. Their structure and their access mechanisms are all 2D. I didn't talk about this, but we have an open TIFF-based format called OME-TIFF. NIFTY is a TIFF-based structure as well. But this is true in the medical imaging world, DICOM, BIDS, etc. These are incredibly well established and supported, and that's great. But what if you want to look, for example, from an orthogonal angle? What if you want to look at a time-lapse mode? What if you want multi-resolution support? Those things aren't well done. Now, many of you may have heard of a structure called HDF5. So Big Data Viewer includes HDF5. There's other implementations of these so-called block-based access, which allows you to come in from different directions. For example, to have orthogonal views. And these are actually fairly well used. These do not work very well in high-performance computing environments, for example, in deep learning. And if you want, I can get into technical details. Each of them end up being yet another proprietary file format. And a critical point, and this gets very technical very quickly, they don't have the flexible metadata that is needed in a lot of the new applications. And the data storage technologies that they can be used on are so-called file systems. So I'm going to try not to get into the weeds here. File systems are the kind of things that are sitting in your laptops, probably in your workstations. They're very powerful and they're basis for an awful lot of what they do, but they are very expensive and they don't scale well. And when I say don't scale, I mean to the multi-petabyte range, which is where we're going to need to go. Many of you will have heard about doing data processing in the cloud. And these cloud resources use a different technology called an object store. And again, I'm going to avoid the technical details, but object stores don't provide good support for very large binary structures like we have in imaging. And that's because when you access a piece of information in an object store, file systems allow you to sort of move through the file and find your reference points. Object stores don't do that. They're cheap and they're scalable, but what they say is, oh, you want this file? Here's the whole file. That works great if you're dealing with Word documents, but if you're working with a 10 terabyte file, if you just say, I just want one plane from a file, and the file system says, that's fine, I'm just going to give you these 10 terabytes, and it's your problem to sort out, that obviously is not going to work very well. The good news is in the last couple of years, several projects have emerged that are building the file format technology to handle this type of storage mechanism. So projects like ZAR, N5, and TileDB. And so you may have heard of these, you may not have, but these are really exciting technologies. And most importantly is that they provide a way for us to think about changing completely how we work. So we've taken bioformats, because bioformats is a way of recognizing all these data, and we've built a library called Bioformats to Raw, which will convert to ZAR, to N5, or TileDB. Now, for those of you who still want TIFF-based formats, we have another library which will do that. But we now have a way to get these data into these formats that will work in cloud-based resources. And we've built effectively a lot of the technologies that you will need for handling the multiple scales, so parameterable, parameterable storage mechanisms, so you can sort of zoom in and out. We have ways of storing masks and labels, and we're starting to move into domain-specific representations, like, for example, in high-content screening. This is very much a work in progress. And the URL that you should see about what the specification looks like and all of that is at the bottom of the slide. One of the results of this, and I think this is probably the most important thing, is that imagine some very large multi-terabyte file that your data is stored in. And imagine you're wherever you are, and I'm here in Dundee, and we want to share that data. These new technologies allow something called streaming, and you're probably very familiar with streaming because it's what Netflix and YouTube and many others use. They don't dump the whole file down the wire and drop it on your laptop. They actually provide access to small bits of data and feed it to you as you need it. And that's exactly what these new file formats do. So it provides a way of not only storing data in the cloud, but accessing data in the cloud. So what we see happening, in this diagram, you sort of see, I think, what the current state of the art is. And so you have a microscope writing out formats and data in whatever formats, and we use bioformats as this on-the-fly translator, for example, from Fiji or many others. But what we think is emerging is a one-time conversion, for example, to a ZAR file, excuse me. And our code name for this project is NGFF, so next generation file format. That could be stored locally, but that could also be posted to the cloud in, for example, these object stores, which are relatively cheap and affordable and scalable. That data would then be available to you, or it could be streamed to applications that can handle that. One of them is Napari. This is an open access, an open source project. So that streaming access to these data sets is now possible. We can make OMERO support exactly the same thing. And actually, this is now all working. And so these data sets that we have example data sets that are in IDR, which now are available in this cloud-based format. This has been kind of interesting. We announced this on Twitter. And so we took the COVID data sets that I showed you earlier and put them in these formats on an S3 resource at EBI. And a few minutes later, basically, we saw people posting saying, "Oh yeah, I've already accessed it, and look, this actually works," which was extremely exciting. And so I think what we're trying to get to is changing the way we think about accessing data across the world of at least the life sciences informatics, certainly in bioimaging informatics. People have generally thought about file download, so downloading everything to my workstation or my laptop, and then I use whatever tool that I like to use to access that data locally. Now we actually have a scalable way of making those data available in the cloud and not forcing people to download the increasingly large data sets. And so all of this is very much a work in progress, and there's an awful lot to do here. But the proofs of concept are all public. I'm very happy to share URLs if you want. If you're interested in this, check out what has become the definitive forum for the bioimaging community called image.sc. You can search for next generation file formats, and you will find not only the outline of what we're doing, but also discussion groups and regular meetings, and all of that is happening and available. And here's our dream, and this is what we dream out in the OME project. What we dream about at night is to take these, is to continue to work with the proprietary file formats from the commercial community or wherever they're coming from, but do a one-time conversion, for example, to ZAR, or there are other structures that are candidates here. Currently, we're putting a lot of effort into ZAR. And so that's a one-time conversion. These files are then open, they're accessible, and they can be, and they're also scalable. I would just, a big caveat is that the proofs of concept that we have done focus on the binary data. That's the thing that people are most interested in. But as you probably know, as soon as the binary data problem starts to look to be solved, people start thinking about the metadata, the experimental metadata, the acquisition metadata, the analytic metadata. All of that has to be supported. We are working with a large number of people in the UK, our colleagues at Riken in Japan, Suzanne Kunis at Osnabrück, David Grunwald and Katerina Strambio de Castilla at UMass in the United States, several others, to build the metadata concepts and to represent them in a truly scalable way in these same file formats. We're just literally starting a 3-year project to do that, so it's going to take a while to get there, but I think we will. So I apologize for going a little bit over, but I hope that's given you an idea of what we've been doing in the OME project and where we're going. So in one slide, I showed you information about file formats and bioformats. OMERO is a data management system, and the IDR, what we're doing about publishing these data sets and annotating them and integrating them, the way we're linking the various resources. Maybe just a take-home message that we can talk about. We think that making bioimaging data fair is possible. We think it's important, and we think that we have the first examples of it being actually scientifically useful and valuable. And I think I just like to finish on this note of that one of the things that's most been heartening and we've enjoyed is that everything I've shown you today is not just the work from my group, but really coming out of a connection to a global community of technology projects and open source technology projects and developers. It's an amazing group is very, very committed to this, this way of working. Many of you will have seen the appearance of national imaging networks. Here in the UK, we have Bioimaging UK, but many of you will have seen examples of that in your own communities. And in particular, those national communities starting to work together, for example, in global bioimaging, and really to define best practice and how we should think about running and building and running these larger resources. So that's really been an awful lot of fun, and probably an unexpected but certainly quite gratifying outcome of all this work. So it just leaves me to thank everybody involved in this. And so we've been quite generously funded here in the UK by the Wellcome Trust, the BBSRC, recent awards from the Chan Zuckerberg Initiative, and several EU awards. And we're very grateful for all of that. We're just starting, as I mentioned, a Wellcome trust-funded project, but also projects with colleagues in the NIH. These are all focused on a lot of the new data types in spatial profiling. Happy to talk about that more. An amazingly talented team here in Dundee. I'd say here in Dundee in quotations because they're actually quite distributed. We've been using remote working now for a long time in OME. And several really great collaborators. I really have to thank Alvis Brasma and Uja Sarkans at EMBL EBI, who've been part of the IDR project from the beginning and the arranging access to embassy. I didn't talk too much about the work with Norio, Suzanne, and Katerina, but this is really around the metadata question I mentioned at the end that will all be coming. And I will just stop. I hope that was comprehensible. I hope some of you are still there. And I'll just ask any questions.